Good morning, everyone. Welcome to today's presentation. I'm Dr. S.K. Singh, holding a Ph.D. in this field. Today, I'll be talking about the qualification of photo stability chambers. We'll explore what these chambers are, how they work, and why their proper qualification is essential. I'm excited to guide you through this topic and share some valuable insights. Let's get started. Let's start by understanding what a photo stability chamber actually is. It's a specialized piece of testing equipment designed to expose pharmaceutical substances and products to controlled levels of visible and ultraviolet light. The main goal here is to assess how sensitive these products are to light-induced degradation, which can affect their safety and efficacy. Now, why do we need this? The primary purpose is to generate photostability data that support key decisions about shelf life, packaging, and labeling. Essentially, it helps us predict how products will behave over time when exposed to light. It also helps us identify the specific degradation pathways that might occur under light exposure, whether for active pharmaceutical ingredients or finished products. Moving to regulatory drivers. These are crucial because they ensure our equipment measures and delivers the correct light exposure as specified by international standards, specifically ICH Q1B during regulatory inspections. Demonstrating that the chamber is suitable for light-induced degradation studies is essential. It reassures regulators that our data is reliable and our testing methods are compliant. Finally, from a scientific point of view, it's important to recognize that inaccurate or uneven lighting can invalidate the stability data we collect. If the lighting isn't consistent, our conclusions about shelf life could be wrong, which could have serious implications for product safety and efficacy. So, in essence, a photostability chamber is a vital tool that helps us ensure our pharmaceuticals remain safe and effective throughout their shelf life, all while complying with regulatory standards and supporting sound scientific research. Let's take a closer look at the regulatory foundation for photostability testing, which is based on ICH Q1B. This guideline is an important international standard that outlines the requirements for testing how drug substances and products respond to light exposure. It's essential because it helps ensure that medications remain safe and effective throughout their shelf life. The core guideline, ICH Q1B, defines specific testing criteria. One key aspect is the minimum visible light exposure, which is set at 1.2 million lux hours. This means that products need to be tested under conditions that simulate prolonged exposure to light that would be encountered during storage and use. Additionally, there's the near UV exposure requirement, which is a minimum of 200 watt-hours per square meter. This ensures that products are also tested against ultraviolet light, which can cause degradation or changes in the drug's stability. Meeting these criteria is crucial because it guarantees that pharmaceutical products are adequately challenged by light, helping us predict how they will behave in real-world conditions. This way, we can safeguard the quality and efficacy of the medicines we develop and distribute. Let's take a closer look at the qualification life cycle, which follows the well-known DQ, IQ, OQ, and PQ process. First, we have the design qualification, or DQ. This is where we confirm that the chamber's features and specifications meet all regulatory and ICH requirements. It's like ensuring the blueprint aligns with all necessary standards before moving forward. Next is installation qualification, or IQ. Here, we verify that the chamber has been properly installed and that its initial configuration is correct. We want everything set up correctly from the start. Then comes operational qualification, OQ, where we test the chamber's functional parameters under defined conditions, such as lighting and temperature controls, to ensure it operates as intended. Finally, performance qualification, or PQ, demonstrates that the chamber can consistently perform under real-world load conditions, confirming it will reliably meet its intended purpose. Overall, this life cycle helps us systematically validate each stage, ensuring our equipment is compliant, properly installed, functioning correctly, and performing reliably throughout its operational life. Now, let's discuss the design qualification, or DQ, which is a crucial step in ensuring our chamber meets all necessary standards. The main objective here is to verify that the chamber we've selected aligns with the ICH Q1B specifications, as well as our own corporate requirements. When we talk about key checks during the DQ, one of the first things to consider is the type of light sources. We have two options. Option 1 involves cool white fluorescent lights combined with near-UV lighting, while option 2 is xenon arc lighting. Choosing the right light source is essential because it impacts the chamber's performance and compliance. Another important aspect is whether the chamber supports measurement and logging of lux hours and UV energy. This capability is vital for tracking exposure over time, ensuring consistent quality and compliance with testing protocols. We also need to ensure that the data acquisition system complies with GMP data integrity standards, such as 21 CFR Part 11. This guarantees that the data collected is secure, accurate, and meets regulatory requirements. As for deliverables, we expect to have a comprehensive user requirements specification, or URS, along with detailed system specifications. Additionally, vendor certifications are essential to confirm that all components meet the necessary standards and specifications. In summary, the design qualification helps us systematically verify that the chamber's design aligns with both regulatory standards and internal quality expectations, setting the foundation for successful validation and operation. Now, let's talk about the installation qualification, or IQ, which is a crucial step to ensure everything is set up correctly. The main purpose here is to confirm that the chamber has been properly installed and is ready for operation. We want to make sure that all components are in their correct places and functioning as they should. Typically, during IQ checks, we verify the chamber's location and utilities. This includes checking the electrical connections and grounding to ensure safety and proper operation. We also verify that all delivered components, such as light sources and sensors, are present and correctly installed. Additionally, initial calibration status of light and UV meters is reviewed. This helps us ensure that these measurement devices are accurate from the start, which is critical for the quality and consistency of your processes. All of these checks are documented thoroughly in the IQ protocol and checklist. These documents serve as a record that everything has been verified correctly and provides a reference for future validation activities. Overall, IQ is about establishing a solid foundation for the chamber's reliable operation, aligning with the overall goal of quality and compliance. Now, let's talk about operational qualification, or OQ. The main goal here is to confirm that the chamber functions as it should across all the key operational parameters. Essentially, we want to make sure everything is working correctly before moving forward. One of the critical tests during OQ is checking the light intensity output. We verify both visible light, measured in lux, and UV energy outputs, making sure they meet our target set points. This ensures the chamber provides the correct lighting conditions for our processes. Next, we have sensor calibration verification. It's important to compare the readings from the built-in sensors against external traceable meters. This step helps us confirm that the sensors are accurate and reliable. Temperature control is another vital aspect. We need to ensure the temperature stays below approximately 30 degrees Celsius. This is especially important to isolate the effects of light, in accordance with ICH Q1B guidelines, which focus on photostability testing. Finally, we validate the control systems and alarms. This includes checking that timers are functioning correctly, alarms trigger as they should, and data logging systems accurately record the operational data. All of these tests together help us confirm that the chamber operates as intended, ensuring consistent and reliable results in our processes. Let's take a closer look at the OQ mapping and acceptance example. Here, we use a light mapping grid, which involves dividing the sample space into a grid. This could be a 3x3 or 4x4 layout. The idea is to systematically record information at each point in this grid. By doing this, we're essentially creating a structured map of the sample space, making it easier to analyze and interpret. This approach helps us visualize how different regions behave or respond, which is crucial for understanding the overall quality or performance. Remember, the key is to be thorough yet efficient in capturing data at each grid point. This method ties back to our larger goal of ensuring quality and acceptance by providing a clear, organized way to evaluate the sample space. Now, let's talk about performance qualification, or PQ. The main purpose here is to demonstrate that the chamber consistently maintains its performance when we load it with real samples, and that it meets the necessary exposure requirements without fail. Essentially, we're verifying that the chamber performs as expected under actual operating conditions. For typical PQ actions, we start by using calibrated lux and UV meters. These are placed right at sample level to get precise measurements of the light and UV exposure the samples would receive. We then run the exposure process until we reach specific cumulative thresholds, which are 1.2 million lux hours and 200 watt-hours per square meter. These values help ensure we're delivering the right amount of exposure over time. Finally, it's important to record all the data related to exposure over time. This data is then reported to confirm that the chamber's performance aligns with our standards. Overall, PQ is a crucial step to validate that our chamber can reliably produce consistent results, ensuring the quality and safety of our products. Let's walk through a simple example of how we might calculate light exposure. This slide highlights the key points involved in that process. First, we need to consider the duration of exposure, meaning how long someone is exposed to the light source. Then, we look at the intensity or brightness of the light, which directly impacts the amount of exposure. It's important to understand that these two factors, duration and intensity, work together to determine overall exposure. For instance, a brief exposure to very bright light might have a similar impact as a longer exposure to dimmer light. This example helps illustrate the practical application of the calculation, which is crucial for assessing potential effects on health or safety. Connecting this to our overall theme, understanding these calculations allows us to better manage and optimize light exposure in various environments. So, by carefully measuring and calculating these factors, we can make informed decisions to protect well-being while still achieving our illumination goals. Let's now move on to some important considerations around alarm and data integrity. First, critical controls are essential to ensure everything runs smoothly. We need continuous monitoring and alarm generation to catch any deviations in key parameters like lux, UV, temperature, or if there's a system failure. This way, we can respond promptly to any issues. Additionally, data logging must be compliant with FDA 21 CFR Part 11, which means maintaining an audit trail and evidence that data hasn't been tampered with. Now, for best practices, I highly recommend using calibrated external meters to verify and cross-check your measurements regularly. This helps ensure your data remains accurate and reliable over time. Moving on to temperature monitoring and control. Although temperature isn't always the primary factor in photo stability, it's still critical because ICH Q1B requires keeping the temperature at or below 30 degrees Celsius during exposure. This is to prevent any thermal degradation bias that could skew your results. For example, during a typical test, the start-of run average temperature might be around 28.5 degrees C, and at the end of the run, it might be about 29.1 degrees C. The temperature variation in this scenario is just about plus or minus 0.6 degrees C, which is well within acceptable limits. And don't forget, always document your temperature logs alongside your light exposure metrics. This creates a comprehensive record that supports your data integrity and helps with troubleshooting or audits down the line. Let's talk about documentation, which is a crucial part of ensuring quality and compliance. When it comes to qualification, there are several key documents you need to have in place. These include the DQ, IQ, OQ, and PQ protocols and their corresponding reports. These documents help us verify that equipment and systems meet all specified requirements at each stage of qualification. You also need calibration certificates for sensors measuring lux, UV, and temperature. These certificates prove that your sensors are accurate and reliable. Mapping grids and exposure logs are also essential. They help us track where and how exposure occurs, ensuring proper documentation of processes. Additionally, standard operating procedures, or SOPs, for operation, maintenance, and requalification are vital. They provide consistent guidance and help maintain the integrity of your processes over time. Now, from a regulatory perspective, it's very important that all qualification documentation is audit-ready. This means it should be complete, organized, and easily traceable, aligning with GMP and QMS requirements. In essence, your documentation isn't just for internal use. It's also your proof during audits that your processes are compliant and well-controlled. So, thorough, well-maintained documentation is fundamental not only for quality assurance, but also for meeting regulatory standards and building trust in your systems. Let's wrap up with some key takeaways. First, having a qualified photo stability chamber is essential. It ensures that the light exposure is accurate and reproducible, which is crucial for meeting regulatory requirements like ICH Q1B. This means your testing conditions are consistent and reliable. Second, following structured IQ, OQ, and PQ protocols is fundamental. These protocols help verify that the chamber is installed correctly, operates as intended, and continues to perform over time. Setting clear acceptance criteria during these phases makes sure everything stays within specified limits. Third, it's important to use calibrated instruments when measuring lux and UV light. Proper calibration guarantees the accuracy of your measurements. And of course, maintaining thorough documentation throughout this process is vital. Good records are key during inspections and audits, demonstrating compliance and protecting your product's integrity. Overall, integrating these practices ensures your stability testing environment is robust, compliant, and reliable, supporting the overall integrity of your pharmaceutical products. For further support, please don't hesitate to reach out to us. We're here to help with any questions or additional information you might need. Simply contact us through the provided details. And with that, I want to sincerely thank you for your time and attention today. Your engagement is truly appreciated, and I hope you found this presentation valuable. Remember, we're always here to support your efforts moving forward. Thank you once again.